The brain reward system consists of several regions, including the hippocampus, the ventral tegmental area, or VTA, the amygdala, the ventral pallidum, the nucleus accumbens, and the prefrontal cortex. Let's focus on the ventral tegmental area and the nucleus accumbens. The ventral tegmental area sends signals to the nucleus accumbens via the axons of dopaminergic neurons. A tonic firing pattern involves signals timed at roughly equal intervals. It has been experimentally shown that tonic firing between the VTA and the nucleus accumbens results in normal interactions of rodents within social defeat paradigms. In contrast, phasic firing patterns involve short spurts of multiple action potentials, followed by long pauses between firing. It has been experimentally shown that phasic firing between the ventral tegmental area and the nucleus accumbens results in rodents exhibiting symptoms of depression in the social defeat stress model. How were the different behavioral tendencies based on different patterns of firing between the ventral tegmental area and the nucleus accumbens shown experimentally?
Optogenetics!
What is optogenetics? Optogenetics is based on the shining of specific wavelengths of light in order to open light-sensitive cation channels in genetically modified cells. This is a powerful tool that can be used to manipulate specific groups of neurons. Two examples of light-sensitive channels used in optogenetics are CHR2, or channel rhodopsin, and halorhodopsin. Channel rhodopsin responds to blue wavelengths, while halorhodopsin responds to greenish-yellow wavelengths. Channel rhodopsin is generally excitatory, and halorhodopsin is generally inhibitory. How do we get these light-sensitive channels into specific groups of neurons? We start off with a Th-Cre transgenic mouse. Th stands for tyrosine hydroxylase, which is one of the enzymes in catecholamine biosynthesis. Its job is to convert tyrosine to dopa, which is then converted to dopamine. Tyrosine hydroxylase is found in the brain reward system, specifically in the mesolimbic pathway, which is the connection between the ventral tegmental area and the nucleus accumbens. A Th-Cre transgenic mouse is a mouse that was genetically modified in such a way that any cell expressing Th also expresses Cre recombinase, which is normally not found in mammals. The second essential optogenetic component is a viral vector. An ADENO-associated virus, or AAV, doesn't cause disease, kindles only a minor immune response, and makes a fantastic gene therapy vector due to the fact that it does not integrate. In other words, it remains in an extrachromosomal state. AAV2 is a serotype of AAV that is strongly neuron-specific. The virus carries a genetic sequence which we desire to input into the experimental animal. Upstream of the protein coding region is EF1 alpha, a general, non-cell specific promoter which will promote the expression of anything that follows. Downstream of the promoter, and on either side of the target gene, are LOXP sites. In the presence of Cre recombinase, they will flip the target gene sequence so that it will be translated backwards relative to its original configuration. The genetic material is said to be floxed if there is one set of LOXP sites on either side of the target gene, and double floxed if there are two sets. LOXP sites also don't exist in mammals. The target gene is originally backwards, so if it ends up being transcribed with no Cre recombinase around, a nonsensical protein will be produced. The whole point of Cre-Lox is to flip the gene, but only in specific cells. ITR stands for inverted terminal repeats. These aid in concatenamere formation in the nucleus, which occurs after single-stranded vector DNA gets converted to double-stranded DNA. WPRE stands for woodchuck hepatitis virus post-transcriptional regulatory element, and it increases transgene expression from a variety of viral vectors. Basically, Cre recombinase is only made by Th-producing neurons, so we can target the ventral tegmental area to nucleus accumbens pathway. CHR2 is produced and expressed only by neurons in the ventral tegmental area in the mesolimbic pathway. So, we injected a virus with CHR2 into the rodent's brain and implanted a blue light there as well. Thanks to Cre recombinase, functional protein only gets produced in the ventral tegmental area. Hence, only the ventral tegmental area will have light-sensitive channels. That means that we can flick on the light whenever we want to make the ventral tegmental area neurons fire. Mwahahaha. This gives us the ability to produce tonic or phasic firing patterns in order to do behavioral tests and assess whether the rodent displays symptoms of depression. This video is based on this awesome article, so you should read it to learn more.